So today we'll discuss about the isolation of bacteria in pure culture. First of all, what is the microbial culture? Purity will come if the culture is done of the bacteria. So microbial culture is a population of bacteria which is grown in the laboratory, and it is a fundamental and the basic diagnostic method for any further step of like diagnosis of the disease and to study the pathogenicity of the bacteria. It is done, and while doing culture, we know which type of the organism is associated with any disease and what is its concentration in the sample, or both can be diagnosed. And the sometimes what happens when we culture the bacteria from any sample, some contaminants may also be present in the bacterial culture, and which may not be of our interest to isolate. And how we culture can be done on the solid media or semi-solid agar media or in the liquid media. And to maintain the culture we need to transfer it periodically, which is known as subculture, to new media to keep the bacterial population live and growing. So pure culture, it is also known as exenic culture, and it contains of a single bacterial species which may originate from a single cell or single organism, and these cells are a clone of us one another. And so the pure culture can be defined as a population or clone which is derived from an individual cell, and it should be free from other contaminating microorganism. And how we can get the bacterial pure culture? By using aseptic technique. And it is the foundation of the all research in the infectious disease. Means we can, after getting only the pure culture, we can further study the bacteria and its properties. And so why we need the pure culture? It is needed for the taxonomic identification. What is taxonomic identification? Means the recognition of or the identity of the bacteria, its essential characters. What is the essential characters of an organism, and what type of the isolate it is. And any scientific study of the organism or pathogenic organism with the objective to characterize the bacteria, pure culture is needed. And what study we can do? We can study its ecology, its morphology, its physiology, and many more things we can study if we get the pure culture. Next one is the diagnostics of a pathogen. Means how this bacteria is associated with a disease condition, that can be studied if we have pure culture. And the clinical presentation of a disease, or what manifestation of an bacteria will show to cause a disease, it can be established by having a pure culture. Next one is the virulence and the pathogenicity studies. Virulence, what is it is? Virulence is the ability of the organism to infect the host and to cause the disease. And virulence is as virulence is also associated with the virulence factors. And what is the pathogenicity study? Pathogenicity is a quality of being pathogenic. Means the bacteria is pathogenic or not. We can study only we have the pure culture. And next one is the elucidation of the metabolic properties. Means the explanation is, elucidation means explanation of the metabolic properties. Means how the, if we have the pure culture, it we can explain how bacteria will behave in the host system, and how it will act to obtain the nutrients from the host. And all these can be studied under the bacterial metabolism. Antibiotic sensitivity test we can do to know the antibiotics against which the bacteria is susceptible. And by knowing this property, we can choose the antibiotic that will be most effective for the treatment of the disease. And so the appropriate treatment can be planned by doing the susceptibility test. And genome sequencing. To characterize the pathogen in the clinical sample, genome studies is done, and genome is a entire complement of the genes which is contained in the DNA of the chromosome. And by knowing its genome sequence, we can further study the bacteria. Next one is the proteomic study. Proteomic study is to study the protein profile of the bacteria. And by knowing the protein profile of the bacteria, we can study how pathogenic bacteria adapted to the antibiotics, different antibiotics, and we can plan for a new target for to act the antibiotics. Also, we can study the virulence mechanism, how any bacteria is virulence, what mechanism it involves, and also for the strain deposition in the microbial collection, which is further needed for the research and for the teaching. And microbial collection provides an authentic biological material for the research purpose. And so only the pure culture can be collected in the microbial collection center. Next is the purity of the culture. How we can define that any culture is pure or not? We adopt some common techniques in lab, and it is purity of the culture is required for the success of the any microbial identification. Without having a pure culture, we can not clearly identify the which type of the bacteria it is and what is its characteristics. And why purity is needed? Sometimes it happens that contaminants which are associated with the target bacteria may be present in the culture plate, and unknowingly they can be subcultured with the chosen organism. So the we should select a such media which prefer on which the all type of the bacteria can be grown. So the non-selective media will help in the identification of the contaminating bacteria also. And how we can know the it is culture is pure or not? If the we see the culture plate, you know that on the agar media on which culture is grown, and we see the same cultural characteristic if the culture is pure. Here you can see, here two different types of the colonies are seen. So the if the culture will be pure, these two different type of the colony will not be seen. If the culture is pure, only one type of the colony with the similar type of the morphology will be seen. And the still when we stain, the all the cells should have a similar type of the form, its morphology and the size should be almost same, and the same staining properties should be there. Means if the bacteria is gram negative, then all the from the anywhere, if we take the culture from the plate and stain, it should show the same type of the staining, and it should give all the culture should give the similar biochemical results. Here you can see this plate and this plate. Here see the type of the culture. You can see various type of the culture. Some colonies are large, some colonies are small, some are pigmented, they are showing yellow color, some are showing brown color, some are cream color. Different colors of the colonies and different sizes are seen. So by seeing the plate, you can say this is a mixed culture. And here you see only one type of the colony which is having almost same morphology is seen here. And when we stain from this plate, different type of the staining or different type of the bacteria will be seen in the same slide. Some may be gram positive, some may be gram negative. Some of the bacteria may have the shape of cocci, means they of the or some may have the bacilli rod-shaped bacteria. When we stain from the pure culture, all the cells in a slide will have the same morphology. Either it may be of cocci or maybe of rod shape. Here you can see all the bacteria which is seen here, it is of same type. Here also you can see. So if we stain from the culture from the pure culture, same type of the bacteria and same type of the staining will be seen. If we stain from the mixed culture, different type of the bacteria with the different morphology and the different stain characteristics will be seen. So how we can obtain the pure culture? Generally, when we first isolate the clinical samples, in some of the cases, mixed culture may be present. And so the from the first plate, which is known as isolation plate, samples are diluted gradually so that the individual organism or the coliforming unit gets separated on the agar surface. And from that, one single type of the colony is picked and again transferred to the new sterile medium. And after the incubation, if the from one single colony we take, then all the organism in the new culture will be from the same organism and it will be pure culture. So how we can dilute and get the single type of the colonies? There are different methods, and different methods are plating, and different type of the plating like the strict plate method, pour plate method, spread plate method can be used. Next one is the serial dilution. Another one is the single cell isolation, and some another method is the enrichment method. In some special type of the treatment is done, specific media is added, or some antibiotics is added, or some UV treatment is done, or sometimes the temperature may be elevated, which may suit for the target bacteria but not to the other contaminating bacteria. So first of all is the streak plate method. This is the most common method which we generally employ in our lab, is the streak plate method. And in this method, at the beginning of the streak pattern, confluent growth, heavy growth will be obtained, and when it is serially diluted uh, by hitting the inoculation loop, and the end of the pattern, discrete single type of the colonies are obtained. Here you can see this is a culture plate, and from a inoculation loop, culture is taken from another plate, and it is streaked like this. Different patterns of the streaking can be done, and if we streak, again we hit the loop, again at the perpendicular we streak the second, again then we again hit the loop, and again take the streaking, and the so gradually the culture is getting diluted, and the last is we can get the individual colonies. Here you can see same, first is streaking, second is streaking, third is streaking, and this one is the fourth streaking. So here you can see the single colony, and from this single colony, after the incubation, one colony is picked and again streaked on the another plate to get the pure culture. Next one is the spread plate method. In this sample, first serially can be diluted, and then the serially diluted sample, they are spread over a solidified agar medium plate, and with the help of a thin L-shaped glass rod, which is known as a spreader, as a thin layer. And another method is the pour plate method. In this also sample can be diluted, and the diluted sample are placed on the center of the petri dish, and upon which molten agar is poured and mixed well to get and allowed to solidify. And after incubation, the colonies will be formed on both on the agar surface as well as in the subsurface, that is in the agar. Here you can see the pictorial explanation of the spread plate method and the pour plate method. Here in spread plate method, sample is pipetted on the surface of the agar. Solidified agar plate is taken on which the sample is spread, and it is mixed with the or spread with the spreader. Then it is incubated, and on the surface colonies are formed, and from this single colony is picked again on stripped on the separate plate. Here in pour plate, first sample, diluted sample is placed on the petri dish. Then molten agar is poured on the plate. Then it is mixed and allowed to solidify. Then it is incubated, and the colonies will be formed on the surface as well as on the subsurface. And the typical colonies from the surface is obtained and stripped again on the separate plate to get the single colony or the pure culture. Next one is the serial dilution method. In this, some of the bacteria which may not grow on the solid media and they grow only on the liquid media. For such type of the bacteria, serial dilution method may be practiced, and the sample from which we need to isolate the bacteria, first it is diluted serially in a sterile liquid medium, and large number of the sterile test tube containing the liquid media are inoculated with the each dilution, and successively in the last tube it is assumed that it will contain the single microorganism which is used as a pure culture. Here in a flow diagram you can see, suppose the it is assumed that in 10 ml medium some culture is added which may contain the mixed culture and having the large number of micrograms. Suppose it is 1000, then from this 1 ml of the culture is removed, again added to the 9 ml of the medium. Then again from that medium 1 ml is removed, and again it will be added to next sterile 9 ml of the medium. Gradually in the last, the inoculum will contain the one microorganism which is used as a pure culture. Next one is the single cell isolation method. In this method, a single cell is picked up from the mixed culture and it is allowed to grow to obtain the pure culture. And it is of two types. One is the capillary pipette method, and in this method, many small drops of the diluted culture medium are put on a sterile cover slip, and then one drop is studied under the microscope, and it is found that the drop which contains only one microorganism is again removed from a sterile capillary pipette and it is transferred to a fresh medium, and then which is allowed to grow as a pure culture. Next one is the micro manipulator method. This uses a microscope along with a micro manipulator, and in this also a single cell is picked from a mixed culture, and it works in association with a microscope to pick a single cell, particularly a bacterial cell, from a hanging drop preparation. And the hanging drop of diluted culture are placed on a special sterile cover slip by a micropipette. And then again in this method also the hanging drop is searched which contains only one microorganism, and this cell is picked up from the micropipette by gentle suction and placed on a large drop of a sterile medium which is placed on another sterile cover slip, and so the drops are collected, and these when the drop contains the more number of the cells, then it is transferred to a suitable media, and which is used for the growth of a pure culture of the microorganism. This picture show the capillary pipette method, and this one is the micro manipulator. Here you can see droplets containing the microbes are put on a sterile cover slip with a capillary pipette, which is observed under a microscope, and the droplet which contains only one is picked up by a capillary pipette, and this is transferred to the culture tube which contains the suitable media for the growth of the bacteria. In micromanipulator method, same type of the method is used, but with the help of a micromanipulator, and and this is transferred to the culture plate for the growth. Another is the enrichment culture method. It is the in this method is suitable when a small number of the bacteria is present in any sample or have a slow growth rate. So this technique provides a specially designed culture environment to the flare up the growth of the target bacteria or the desired bacteria, and this makes the condition favorable for the growth. And by providing some ingredients in the media or some environmental conditions, it unfavor the growth for the competing organism, and the mixed population bacterial population are inoculated in a medium which is having a defined but limited composition. Chemical composition means the known chemical composition, and allowed the bacteria to grow under the control condition, means what temperature, on which temperature it will be incubated, how the air supply will be there, light condition and pH, all will be manipulated, and only and this will favor the growth of a particular type of the bacteria, while the other competing bacteria will not grow. So why we need to maintain the pure culture? It is needed to maintain the pure culture for the further studies or to viability of the or the purity of the microorganism, and it should be always, it should be free from the contamination. So the for uh, culture maintenance, pure culture maintenance, we need to transfer the culture into a fresh medium which is known as the subculturing, and it is for the continuous growth or the viability of the microorganism. And culture into the subculture into a fresh medium, we should always practice the aseptic condition. How we can follow the aseptic condition? Like when we transfer inoculation loops should be properly heated and cooled, or the media should be free from the contamination, and in the lab also we should follow the aseptic conditions. And another method to maintain the pure culture is the microbial preservation, and which aims to preserve a microbial strain alive, free from the contamination and without any variation or the mutation, like it's the original isolate, means the which organism we have isolated primarily, that should be maintained as a pure culture for long time. Then only the pure culture will be maintained. So this completes the chapter of the isolation of the bacteria in pure culture.